Okay, so let's get started. So, welcome to week 9 of NGS data analysis and applications. So, last week we talked about RNA sequencing data analysis and how we do uh, differential expression analysis, what are the different caveats to it. Today, we'll be continuing on that front only. So, we'll talk about different models that are used in differential expression analysis, how you can correct for certain multiple hypotheses, and so on. Okay. Okay. So last class also we were discussing about DEC2. So we'll catch up from there only. So basically, in DEC2 is a package in R that is used for differential expression analysis, where the final step in the DEC2 workflow is to take the counts for each gene and fit the model for testing for differential expression analysis. Okay. So yeah, this is your basically your workflow, and the last three uh, modules, basically modeling raw counts for each gene, testing for differential expression, and shrinking the log to fold changes. We'll talk about these three modules primarily in today's class, and yeah, so let's get on. Okay, so this modeling part. Just a second. So this modeling part is actually very important for the differential expression analysis. Depending on the type of model that you choose, you get you'll get your results and your p-values and everything. So the model that are used in DEC2, there are numerous models, like 3-4 models that are used. So we'll look into each of the model and what it does. First, we'll talk about a generalized model, and then we'll look into the different types of model used in DEC2. Okay. So first is the generalized linear model. So the count data that is generated by RNA-seq exhibits over dispersion. So what do you mean by dispersion? Dispersion is basically a statistical term that is used to quantify the ratio between the variance of the data and the mean. So if your ratio between variance and mean of the data is very high, we call that data over dispersed. So what it means, if you think about it intuitively, it's basically if you have a yeah, what it means basically is your data will be your distribution will be more spread out. So your mean is lower, but your data is more spread out. So that is what over dispersion means. So in general, if you look at RNA-seq counts data, so we talked about how you can get counts basically. So the counts is indicating the number of reads that are supporting a particular transcript. So if you plot the distribution for that particular counts data, what you'll see is your data is very well spread out. So like it's more bell shaped, and the bell is more depressed during the in the mean. So basically you'll have something like this, and in the middle it will be more depressed and the spread will be more. So that is what we mean by over dispersion. It's basically the ratio between variance and mean. So the count data generated by RNA-seq exhibits over dispersion, which is the ratio between variance and mean, and the statistical distribution used to model the counts needs to account for this. So this is why I was telling the model that you are using for getting the differential expressions of the genes that you are interested in must take into account that your data initially is not a normalized data. It's more of a negative binomial distribution. It's more spread out. So DEC2 uses a negative binomial distribution to model the RNA-seq counts from this equation. So basically this equation is negative binomial. So this NB denotes the negative binomial distribution, and the mean so how we represent this distribution is basically the mean and the variance. So when we represent normal distribution, we write it as n in the bracket 0, 1. What it means is you have a mean of 0 and a standard deviation of 1. So here it's a negative binomial distribution with a mean which is scaled and basically scaled by a normalization factor, because this mean is taken as normalized counts. So you have a negative binomial distribution with normalized counts as your mean, and whatever the dispersion or the standard deviation for that particular data set is. So, and your k on the left hand side denotes the raw counts for a particular gene in a particular sample. So these parameters, these two parameters basically are required, are the size factors and the dispersion estimate. So the size factor is denoted by this normalized counts and scaled by that factor, and the dispersion estimate, which is alpha I in this case. So once you have this kind of a distribution, a generalized linear model, which is also known as a GLM, of the negative binomial distribution family is used to fit the data. Modeling is basically what it is doing is it is modeling the particular data that you have into a particular distribution, predetermined distribution. So it is mathematically formalized way to approximate how a data behaves given a set of parameters. So you have a set of parameters given, and you have given a data, and you want to see how it behaves. So like fitting the data to a particular model. In this case, the model is a particular distribution. So this thing is known as the modeling. So it is a mathematically formalized way to approximate how the data behaves given a set of parameters. Sorry. Okay. And in statistics, the generalized linear model is a flexible generalization of an ordinary linear regression that allows for response variables that have an error distribution model other than a normal distribution. The GLM generalizes linear regression but by allowing the linear model to be related to the response variable via link function, and by allowing the magnitude of the variance of each measurement to be a function of its predicted value. So I'll not go into details of what is a GLM and everything, but I just wanted to give you an idea how this fitting and fitting of a model to a particular data works like. So basically you will have a predetermined set of notions how your data should look like, and you're trying to fit the data to that particular predetermined notion. In this case, that's a distribution. In some cases that can be a regression or anything else. So this is a special case for that, where you use a negative binomial distribution, and you also have a dispersion estimate, which actually takes into account the fact that the RNA-seq data will have over dispersion, where your variance will be much more than your mean, and the data will be well spread out. So after the model is fit, the coefficients, basically your parameters, the coefficients will be your parameters. The parameters are estimated for each sample group along with the standard error. So in this case, basically the parameters that are estimated are usually estimated by maximum likelihood estimation, or you can use other estimation techniques as well. But yeah, most likely you'll see in many cases MLE is used, which is the maximum likelihood estimation. And the coefficients are the estimates for the log2 fold changes, which will be input for the hypothesis testing part. Is it at all significantly upregulated or significantly downregulated? That part comes into the part of hypothesis testing. Before the hypothesis testing part, you need to generate the log fold to change values, which will be done through this particular generalized linear model, which is a negative binomial distribution. Right? Okay. So next comes the hypothesis testing part. So I'm assuming that you have certain idea about what is hypothesis testing. So basically you'll have two contrasting hypotheses or contradicting hypotheses, and you will try to find evidences for both. And then what you'll do is you put all the evidences in a distribution, and you check if the particular evidence for your particular hypothesis, where in the distribution it is lying. If it is lying at the two ends of the distribution, and typically we take p-value as 0.05, what it means is basically your data is either your particular data point, or your particular hypothesis, and the resulting data from that particular hypothesis is either in the higher 5% to 2.5%, or in the lower 2.5% of the distribution, which means it is not going to be randomly found that easily, because it is present in the 5% of the data set, either in the above, or either during the tailing off. So this is kind of your normal distribution. Either your data is present here or your data is present here. So that is how we do hypothesis testing, and that is how the 5% or the 0.05 p-value comes into play. Okay, you are claiming that you can like your hypothesis is true given the fact that a particular data point which is contradicting your hypothesis is only present in 5% of the distribution. So that means it is highly likely that your hypothesis is true. Okay, so the first step in hypothesis testing is to set up a null hypothesis for each gene. So in this case, the null hypothesis will be there is no differential expression. So yeah, in this case, the hypothesis, null hypothesis is there is no differential expression of the particular gene across the two samples, which is the log fold change is 0. Notice that we can do that without observing any data because it is based on a thought experiment. Second, we use a statistical test to determine if based on observed data the null hypothesis is true or not. As I was saying, so like you have a thought experiment, like you set up a null hypothesis saying that this particular gene is not differentially expressed, but when you see the distribution of the log fold changes and your particular genes across data, so what you see is by random your the log fold change is greater than 2 in most of the cases, and it is less than 2 in only 5% of the cases. It's less than 2 in only 5% of the cases. So that is when you reject your null hypothesis that there is no differential expression and accept the alternate hypothesis is that there is a differential expression. Right. So the steps are, you have a model and you get log fold change values from that particular model. Then you see the distribution of the model and then you do a hypothesis testing on the model. Right. So in DESeq2, the wald test is the default hypothesis testing when it comes to comparing two groups. So what is the wald test? So it is also another statistical technique. So this is a test that is usually performed on parameters that have been estimated by maximum likelihood. As I was saying, in this particular model, you will fit the data into the model and you'll estimate the parameters which are the coefficient of the log fold which basically will be the log fold to changes using maximum likelihood estimation. That's an estimation theory technique to kind of determine particular coefficients and parameters of the particular model. Right. So in this case, the test used for hypothesis testing is wald test, and it is usually performed on the parameters that have been estimated by maximum likelihood. So in our case, we are testing each gene model coefficient, which is the log fold change value, how much upregulation or downregulation has this particular gene undergone, comparing the samples. So so we are testing the LFC, which was derived using the parameters like dispersion, which were estimated using maximum likelihood. If there are more than 2 sample classes within a variable, for example, if you had low, medium and high treatment levels, so basically you have multiple samples you are checking across, so you differential expression is not only limited to like kind of 2 samples, you can like extend it to multiple samples and check if you're like say you can have a set of like 3 your control samples and 3 diseased or 3 treatment samples, something like that. So you can check it for multiple samples and how the log fold change is changing across different samples for a particular gene. If there are more than 2 sample classes within a variable, for example, if you had low, medium and high treatment samples, then DESeq2 will generate 2 pairwise comparisons when low is set as the control, low versus medium, low versus high. So basically it will take one of the sample and it will compare it with the rest of them. So and DESeq2 implements the wald test by taking the log fold change and dividing it by the standard error, which will give you the Z statistic, which is the Z score. Basically you're normalizing this. So the Z statistic is comparable to a standard normal distribution, and p-value is computed reporting the probability that the Z statistic, at least as extreme as the observed value, would be selected at random. If the p-value is small, we reject the null hypothesis and state that there is evidence against the null hypothesis, that is the gene is getting differentially expressed. So a couple of things to keep in mind here. Your null hypothesis states that like you've assumed that in your null hypothesis that the genes are not differentially expressed. After that, you do this wald test and you compare the samples, basically the differentially expression samples that you are comparing against, control versus treatment or different types of treatment or whatever, and you take the log fold change of each of the genes of the particular comparisons, and you divide it by the standard error, which will give you a z scores. And the z score, you get the z scores, you plot the distribution of the z scores, and then you compare this z score distribution with a standard normal distribution, and you compute the p values, and which is basically the probability that the z statistic, as at least as extreme as the observed p value, would be selected as random. So what you are saying is, if your null hypothesis is like that, you do not have any differential expression. But when you see is, maximum of the data that you are seeing in the standard normal distribution is having log fold change. Only 5% of the data, or less than that even, are having no log fold changes in across different tests. So what you can say is, if I pick a data randomly from the distribution, it is highly likely that that particular data point will have log fold change value, which is like not equal to 0. It will be a positive or a negative, whatever be it. If you randomly pick a point from the distribution, it is highly likely that that particular value will have log fold change. And how much likely is it? That is how we quantify the p-value. So if it is at least 95% likely that if you pick a point at random and that will have particular log fold change, is basically 95% of the data will have log fold change and 5% of the data won't have. So that 5% is denoted as the p-value, which is in most cases it is used as 0.05. So if that is the case, that only 5% of the data will not have log fold change and the rest of the data will have log fold change, then you reject the null hypothesis, saying that there is enough evidence against the null hypothesis that there are, in 95% of the cases, the gene will be differentially expressed, and only in 5% of the cases the gene will not be differentially expressed. So that is how you reject the null hypothesis and accept the alternate hypothesis, which will be that there is evidence that the particular gene will be differentially expressed, right? Okay, so that was the wall test. We also have other tests for hypothesis testing, which includes the likelihood ratio test. So this is also a very sorry. Yeah. Other than the wall test, you can also use likelihood ratio test, which will provide you as an alternative to hypothesis testing using the wall test. So just a second. Yeah. LRT serves as an alternative hypothesis test for when we are comparing more than two sample classes. So for example, you have 3 controls, you have 3 samples at a treatment level of a certain degree, you have another set of samples which has a treatment level of another degree, and so on. So when you have more than 2 classes of samples, LRT is something that is commonly used. Rather than evaluating whether a gene's expression is up or down regulated in one class compared to another, the likelihood ratio test identifies genes that are changing in expression in any direction across the different sample classes. So how does this compare to the wall test? The wall test only estimates one model per gene and evaluates the null hypothesis that log fold change is equal to 0. But for the likelihood ratio test, it is also performed, other than the estimating one model for gene, it is also performed in parameters that have been estimated by maximum likelihood. So just like recapping, so in your generalized linear model, in your hypothesis testing part in the wall test, what you are doing is you are modeling you are performing the wall test on parameters that have been estimated by maximum likelihood. But for likelihood ratio test, you are modeling for different sample classes, and according to those different sample classes, you will have different set of parameters estimated. So it is not one model per gene. This will be depending on the number of sample classes you will have. You can have multiple models per gene. Okay, so it is also performed on parameters that have been estimated by maximum likelihood. For this test, two models are estimated per gene. The fit of one model is compared to the fit of the other. So that is what exactly what is it is sounding like likelihood ratio test, which of the model is giving you a better idea. So this is basically your likelihood ratio statistic, and your M1 and M2 are the two different models. So we are evaluating the null hypothesis that the full model fits just as well as the reduced model. If we reject the null hypothesis, this suggests that there is a significant amount of variation explained by the full model, and our main factor of interest here. Therefore, the gene is differentially expressed across different levels. DESeq2 implements likelihood ratio test using analysis of deviance, which is ANODEV. I think you have heard of a variation of this, which is ANOVA. So that is analysis of variance, this is analysis of deviance, and it uses it to compare the two model fits. So basically you will have two sets of opposite models. One model that fitting the hypothesis that the gene is differentially expressed, another model fitting the hypothesis that gene is not differentially expressed, and then you kind of compare these two models to see which is having a better better likelihood to explain the data that you have at hand. So you compare the model fits using an analysis of deviance, which is ANODEV, and it is shown that likelihood ratio follows a chi-squared distribution, and this can be used to calculate the associated p-value as well. So I am not going into details into chi-squared distribution and how likelihood ratio test can be used to calculate the p-values, but yeah, just remember this part ki you're comparing two different models, two different alternate models, and you are seeing which model actually fits the data better. So yeah. So now when you have calculated like likelihood ratio test or Wald test, whatever hypothesis testing you're doing, these are multiple tests that you are performing. Regardless of whether you use the Wald test or LRT, each gene that has been tested will be associated with a p-value. It is this result which we use to determine which genes are considered significantly differentially expressed. However, this p-value that you get from either from Wald test or LRT or whatever model you are using, you cannot use this p-value directly. Before moving on to like how you can use the p-value and how you can correct for the p-value, I just want to recap another time what exactly does the p-value mean. So a gene with a significance cutoff of p less than 0.05 means that there is only 5% chance that it is a false positive. For example, if we take 20,000 genes for differential expression at p-value less than 0.05, which is 5%, we would expect to find another 1,000 genes by chance. If we found 3,000 genes to be differentially expressed in total, roughly 1/3 of our genes are false positive. Right? We would not want to sift through our significant genes to identify which ones are actually true positives. So since each p-value is the result of a single test, the more genes we test, the more inflation we see in the false positive rate. So basically what happens is, the more testing you do, the more your p-value decreases, and the more, but actually technically your p-value decreases, but if you see in the data, you are inflating your false positive rate. Although your p-value is low, but what you see if you look across the data, what you'll find is you'll have a lot of false positive. Why is that the reason? Because for each p-value that is generated, you are testing the gene multiple times. So for multiple times, you are getting one p-value, but you are considering only one across all the testing that you have done. You need to control, or you need to basically correct for that p-value to get a better idea about if it is actually a false positive or actually it is a true positive. Yeah, so since each p-value is the result of a single test or a single gene, the more genes we test, the more we inflate the false positive rate. This is a multiple testing problem. So this is a problem that is not only limited to RNA sequencing analysis. This is something that is present everywhere where you do multiple testing multiple statistical tests, and you get p-values, like trying to get a hypothesis testing. So in all those cases, the number of times you're testing this statistical analysis, the more you are inflating your p-value, and by inflating I'm not meaning it is increasing the p-value, it is actually decreasing the p-value, but the real p-value is much higher than the decreased p-value that you are getting due to multiple testing. So this is something that we need to correct for. So in these cases, we'll have a few approaches for multiple test correction. So out of them, the Bonferroni correction is one of the most widely used. So so the Bonferroni correction, or the Bonferroni threshold that we see, is something that is also used in GWAS. So if you remember from the initial few classes, we talked about genome-wide association and how we do GWAS and stuff. So in GWAS also, so what you do is basically you testing out a phenotype that can be anything with a particular genotype, and you're trying to find if that particular genotype has any association with a particular phenotype. But the point is, you're testing that genotype over and over and over again. So depending on the number of SNPs or the number of genotypes you're having, that is the total number of tests you are going through for a particular phenotype. So the p-value that you are generating will be technically much lower, but in real life that p-value is actually higher, but the technical p-value that you're getting is much lower, giving you a false sense of hope and the false sense of assurance that you have a very statistically significant result, but actually it's not. You need to correct for the number of times you have tested for it. So the Bonferroni correction is a simple way to do that. So it's basically you divide the p-value is by the you multiply the p-value by the total number of tests, and this is a very conservative approach with a high probability of false negatives, so generally not negative and recommended. So basically you will be losing not technically losing, but you'll have a higher number of false negatives, which means basically it was actually a negative, and you categorically kind of associated it with a false, meaning it was actually a negative and then you accepted the negative. Yeah, that is what it means. So basically what it is doing is you are multiplying the p-value by the total number of tests, so that your p-value is actually increased, not decreased. So the threshold that how we use in GWAS, so for example, if I randomly show you the plot, so this is how Manhattan plots look like. This is how Manhattan plot looks like. So it's on your y-axis is basically your log of the p-value change. So basically, if it is a 6 if you take a negative log p and your value is 6, what it means is your p-value is 10 to the power minus 6. So see, we are not considering a 0.05 or a low p-value like that. We are considering, if you see the cutoffs for here, it's around 5 and this is around 7 point something. So 10 to the power minus 5, if the p-value is coming, then only we are considering it as a significant association, otherwise we are not. So why this very low p-value? So that is how we get that is basically, it's basically this p-value 5 into 10 to the power 8, but how we get that is basically you take the normal threshold, which is 0.05, and you divide it by the number of tests. In general, the number of tests done is 10 to the power 6, which is 1 million tests, and you have a p-value of 0.05, and you divide 0.05 by the total number of tests, so that you can get a less inflated p-value and get the cutoff of what is 0.05 p-value meaning in this context. That is how it is done. So for Bonferroni, it is quite simple. You just multiply or divide it by the total number of tests, and this will be a very conservative approach with a high probability of false negative. So it is generally not recommended in this case, but in other cases you'll see Bonferroni correction used a lot of it. And the next thing is FDR, or false discovery rate, or the Benjamini-Hochberg correction. So they defined these two people, Benjamini and Hochberg, defined the concept of the FDR, which is the false discovery rate, and created an algorithm to control for the expected FDR below a specified level of given a list of independent p-values. So that the concept of Q value or the story method comes in. So the minimum FDR or the false discovery rate can be attained while when calling that feature is significant. For example, if a gene X has a Q value of 0.013, it means that 1.3% of the genes that show p-value at least as small as the gene X are false positives, right? So this kind of questions, MCQs might come in your exams as well. So what does this particular value mean? So a Q value, just remember, a Q value means whatever be the value is that percent of genes that show p-value at least as small as that particular gene are false positive. So it gives you an idea about what are how much is the false positive present in the data according after you fit the model, and after getting that you can have an idea about what to take, meaning when to consider a gene to be differentially expressed or not expressed depending on the p-value that you are getting. So this DESeq2 helps reduce the number of genes tested by removing those genes unlikely to be significantly differentially expressed prior to testing, such as those with low number of counts or outlier samples, which you can remove during gene level quality control. You can add a level of quality control on the data, and you can remove this low number of counts and outlier samples, so that it can automatically, the p-value can be corrected by the fact that the number of tests that you are doing after removing those particular genes or samples will be lower. However, the multiple test correction is also implemented to reduce this false discovery rate using an interpretation of the Benjamin-Hochberg procedure. So if you are given a FDR value of 0.05, what does that actually mean? So when you'll be in the next class, we'll be talking about we'll be doing some hands-on on RNA-seq data and analysis. There you'll see the differentially expressed after doing the differential expression analysis, the file that has been generated will have a column of FDR, and it will have a column of p-value and adjusted p-value and log fold change and everything. So there what we do is, if you want to actually check if a particular gene is actually differentially expressed or not, so obviously we sorted by the log fold change values, but also the FDR should be less than 0.05, which means that the proportion of false positives that we expect amongst our differentially expressed genes is only 5%. For example, if you call 500 genes as differentially expressed with an FDR cutoff of 0.05, you expect at least 25 of them to be false positive. So lower the value of FDR, the better result it is for you. The more confident result, I should say, that you can claim that particular gene is not a false positive, claiming to be differentially expressed. Right. Okay. Now comes the part of functional enrichment analysis. So we talked about differential expression analysis. So RNA-seq data, from the beginning we talked about how to align RNA-seq. There are introns, exons and everything. How do you align that? Then how can you get the counts data? How can you normalize the data and everything? Then how can you find the genes that are differentially expressed compared to different samples and different set of conditions? After that, After that, we looked into differential expression analysis and different false discovery rates and log fold change and stuff like that. Now we come to more downstream processes. So you start with the RNA-seq data, you do alignment and everything, you get the counts data, you normalize, you do QC and everything, then you do differential expression analysis, and after that the rest of the downstream processes are mostly functional enrichment analysis. But what is functional enrichment analysis? What does this actually mean? So whether you are measuring mRNA expression, protein expression, DNA methylation, or whatever you're you're measuring, you will likely end up with a list of genes or gene products from which you would like to derive functional relationships. Meaning, like, say for example, you are doing differential expression analysis and you find, say, 5 genes which are highly differentially expressed in the disease sample compared to the control sample. After you've got a list of those 5 genes that are differentially expressed, what would you do with that gene? You would try to find out what are the functions of the genes. Sorry, if those genes are somehow involved in particular cellular processes, or if those genes are involved in some sort of disease pathways or whatever, you would want to have a functional like kind of association between the genes that you have identified to be differentially expressed or whatever you are trying to do, those genes, and what is actually their biological function, to get an idea about, like, if that particular gene is actually differentially expressed, if the level of the gene expression of the gene is elevated, what is the biological process that it will hamper, or that it will upregulate or downregulate, to how will the change in expression of the gene affect the function of the cell or the tissue or whatever you are trying to come up with. Okay, so in omics, functional enrichment is an umbrella term encompassing approaches to derive biological or functional meaning from a set of genes or a gene list. So you have identified a set of genes. Now you want to see how this gene list actually is affecting your particular condition, what pathways it is upregulating, what pathways it is downregulating, or whatever protein it is forming more or less, or whatever. You want to have functional meaning to the list of genes that you have identified. The purpose of this spotlight is to shed light on what we mean by functional enrichment analysis and to point out some methodological limitations and other considerations. Functional enrichment analysis can be easily used to integrate different types of data and identify mechanisms, functions, processes, targets, or regulators of diseases like cancer or other kind of conditions. However, the results are sensitive to the quality of the data going into the analysis, the methods used, selected background genes for specific methods, and the knowledge bases used to inform these methods. So according to this, there are 3 general approaches for functional enrichment analysis. First one is known as over-representation analysis, which is ORA. Next is functional class coding, and the third is the pathway topology. We'll cover each of them a little by little in the further slides. So the first is the over-representation analysis, which is the ORA. Over-representation analysis compares the proportion of genes associated with a gene set found in an input list versus the proportion of genes in a background gene list to determine whether a gene set is over or underrepresented. A p-value is generally assigned using a Fisher's exact test, chi-square test, or a similar statistical method. Over-representation analysis is conceptually easy to understand, but it is limited due to arbitrary thresholds used to define input lists and statistical assumptions such as gene independence, which rarely holds true, because like the expression of one gene. So for example, why are we interested in doing this for functional enrichment analysis? Because the results that you'll get from differential expression analysis will tell you only half the story. Say for example, you have 5 genes that are getting highly expressed. You cannot claim that those 5 genes are the result are the reason for this condition in the sample. That is not the case, because when one gene will upregulate itself, because that gene is part of a network, it's not acting alone, it will be part of a network. Other genes that are associated with this particular gene, their expression will come up or down depending on the expression of this particular gene. So independent expression is a myth. It does not happen in biology. So any kind of gene that is upregulated or downregulated will have some consequence on the other set of genes that is associated with, right? So, and what do we mean by under or overrepresented? So for example, you have done a differential expression analysis and you have identified 10 genes that are present, that are differentially expressed. Now, out of that 10 genes, you want to check which is the pathway that is getting like affected by this upregulation of this particular 10 genes. So what you do is you so first of all, what is a pathway? Pathway is like a collection of genes. In bioinformatics, how we define pathway is a collection of gene sets. So basically, if a particular pathway is having certain elements in the pathway, in the cellular signaling pathway, the proteins that are involved in the cellular signaling pathway are like getting generated by the expressions of the genes. So we kind of take the proxy as the genes only, not the proteins that are involved in the pathway of the signaling pathway, right? So those set of genes, a gene set is defined as a pathway in case of bioinformatics analysis here. So say for example, you check for 5 pathways. Say for example, you are checking for apoptosis pathway, you are checking for say metabolic pathways like glycolysis, oxidative phosphorylation, fatty acid oxidation, other types of pathways. And what you see is that these 10 genes that you have identified to be differentially expressed, 8 of these 10 genes are from a particular pathway. So what it means is that particular pathway is overrepresented in that set of 10 genes that are differentially expressed. So if a particular gene set is over or underrepresented in a particular set of genes, in this case the particular set of genes that you are testing for is the differentially expressed genes. So if a particular gene set is over or underrepresented, you assign you do a Fisher's exact test or a chi-square test, and you assign a p-value that particular gene, that particular pathway that is getting overrepresented in those 10 genes, it is not by chance, it is not random, there is some background to it, right? And overrepresentation analysis, you can do it with pathways and other set of genes and everything. You don't only have to do it with pathways, right? You can do it with a set of genes that are not a pathway but somehow are involved in some mechanism in the cell, something like that. It does not necessarily have to be a pathway, it can be any set of genes. So so these methods are also sensitive to the size of the gene lists, performing much better when the gene list exceeds a size of 50, right? So this size of a gene list is also important when we'll be looking into GSEA, gene set enrichment analysis, and some stuff like that, where we'll try to normalize by the number of genes that is present in a particular gene set or a particular pathway, right? So that number of genes present, a number of input gene lists that you are giving to check if those lists, those genes are over or underrepresented, the number of genes, such genes, is very important. So the performance depends on the number of genes that you will be providing. Okay, just a second. Yeah. So the next method is basically functional class coding. So there are 3 methods, as I said: overrepresentation analysis, functional class coding, and pathway topology, right? So we talked about overrepresentation analysis. Now we'll talk about functional class coding. It includes rank-based methods, which are known as like most common are GSEA and other methods, and these are more sensitive to the ORA methods. So basically, the overrepresentation methods are less sensitive compared to functional class coding. So what is the major difference? In overrepresentation methods, you are only checking if a certain number of genes from a pathway or a certain biologically relevant set of genes is over or underrepresented in a given set of genes, but you are not quantifying it in any form or manner. Like what happens if like 2 separate gene lists are present, but both of them are equally represented in your differential expression analysis? So then what do? You need a quantifiable method to say which gene set is actually more appropriate and more represented in a particular gene set compared to other, right? So these are the functional scoring class, which includes rank-based methods like GSEA, and these are more sensitive to the ORA methods. Rather than supplying a gene list based on an arbitrary threshold such as a log-fold change or a p-value or whatever, these functional class coding methods consider the entire data set, right? It does not only check for the significantly upregulated or significantly downregulated gene. It will check across the whole data set. In rank-based approach, the distribution of genes from particular gene set at the top or bottom of the ranked list ultimately determines the significance of a pathway or a gene set. However, if the user only has a set of genes with no apparent ranking order, and an FCS or a functional class coding is inappropriate. So if you only have a set of genes but you do not have a ranking order of those genes, like which comes after what, or in some way, say ranking might be in the form of like which gene influences the other, like in downstream manner. So if you have a gene set of 1 2 3 4 5 6 7 8, so basically 1 regulates 2, 2 regulates 3, 3 regulates 4, something like that. So that kind of relationship or ranking is important for an FCS approach. But there's another method that comes into hand. So both FCS and ORA discard large amount of information about a pathway. These methods can be more aptly described as gene set approaches. Gene sets are collection of genes formed on the basis of shared biological or functional properties as described by a reference knowledge base. Knowledge bases are database collections of molecular knowledge, which may include molecular interactions, regulation, molecular product, and even phenotype associations. So some of these example of pathways are gene ontology, cake, reactome, wiki pathways, panther, etc., etc. In contrast, a pathway is not a list of genes but rather includes an interaction component usually related to a specific mechanism or a process. As I was saying, like there needs to be an interaction component, or there needs to be a hierarchy or understanding of how these genes interact to carry out a certain mechanism or a certain process. Now on the other hand, topology based methods consider structural information such as gene product interactions, positions of genes, and types of genes within a pathway. These methods, which include impact analysis, topology based pathway enrichment analysis, have shown to produce more accurate results when the user wishes to understand the types and directions of gene interaction and the underlying mechanisms. However, such methods often require experimental evidences for pathway structures and gene-gene interactions, which is largely unavailable for many organisms, so which is very difficult to find. So for human data, you'll find you have databases like String and other databases where you can go and check how a gene is interacting with other, if it is upregulating, if it is downregulating, or whatever it is doing. But for other organisms or rare organisms or organisms that are less studied, in those cases you will not have a database to give you this kind of information. So topology based analysis is not a good idea in those cases. Right. Okay, so these are just, I've listed down the list of tools that are mostly used for these kind of approaches. So GACA is something that is very widely used. So this is the website of GACA, which is the MSigDB website. We'll come back to this. So here, if you want to kind of learn more about GACA and like what it does and everything, this is the paper. This is the paper that is actually kind of tells you what exactly GACA is and like how it performs. So I'll put this in the chat if you're interested. Yeah, you can take a look. It'll be it is actually interesting. So this is a 2000, I think 5 or 7, or like 2005 paper. It's quite old now, but this is GACA is something that is very widely used till now as well. Okay. Let's go back here. Yeah. So these are the list of tools. David is a tool that is also very widely used. So let's quickly go to this is a web-based tool. You can use APIs as well, or you can just do start analysis from here, and you can like submit your gene list some and everything, and you can give your data set and everything, and you can like get the information and everything. So there are a lot of tools that are supported by David. So functional annotation is one of them. So you can play around a little bit if you want for this one. So this is a web-based tool. You do not need to download even. So I'll leave the link here as well. Yeah, and Enrichar is also a tool that is quite commonly used. So Enrichar, it's basically if you give a set of genes, say for example I give PPARG, something like this, PPARG, and I search. mm, yeah, I'm gonna do a submit, and you can see whatever data sets it is part of, what kind of cell types, pathways it is associated with. So say for example Reactome 22 database, you'll have these set of pathways. For other databases, say for example wiki databases, it's part of inflammatory response pathways, it is part of uh, kinase expression pathways, JAK-STAT pathways, and everything. So these are different databases. So KEGG, Reactome, Wiki Pathway, these are like different BioCarta. So these are different databases of pathways and interaction databases, which actually has the information about which genes and which pathways are involved in which particular functions and everything. So you can try to use these particular databases and like play around, like it's fun. You give a random gene name, and it'll tell you which pathways it is associated with and what are the different functions of this. So yeah, okay, let's get back here. Yeah, okay, so now we come to something called as gene ontology analysis, and like what is gene ontology. We'll first learn that, and then we'll look into other types of analysis as well. So there are actually two different entities that are commonly referred as the gene ontology, or GO. So GO, or the GO, is something that is like very commonly used by bioinformatician. It's, it's actually gene ontology. We say GO or something like that. Okay, the ontology itself. Sorry. Okay. The ontology itself, which is a set of terms with the precise definitions and defined relationships between them. So first is what is ontology. So ontology you'll have for genes, you'll have for tissues, you'll have for organs and everything. So I'll quickly show you. Say for example tissue ontology. It's easier to understand. Right. Say for example Brenda. Yeah, Brenda. Say for example I search for a term here, lymphocytes. Yeah. So what it gives you is basically the hierarchy. So the top hierarchy is tissue, cell types, and enzyme sources. Then comes the animal hierarchy, which is basically the human. Then whole body. Then hematopoietic system, which is the blood system. Then blood. Then hematopoietic cell. Then inside it comes leukocytes and lymphocytes. So the hierarchy of the categorization it gives you. So the similar concept is for gene ontology as well. So this is the gene ontology resource. So you'll have these set of ontologies. So for different kind of organisms, you have different ontologies, and I just want to show you. Say for example, I'm not sure if you work or not. No. I think you have to give the ontology term, the gene name won't work. So let's go here. Yeah. So the ontology is like characterized based on molecular function, like molecular level activities performed by gene products, cellular component, where it is present, where it is expressed, where is the gene expressed, and the biological processes, which basically which factor it is contributing to, meaning which biological process, cellular process it is contributing to, and so on. So yeah, this is what I was looking for. This is the gene ontology graph. So basically, just let me remove this. Yeah. The structure of gene ontology can be described in terms of a graph where each gene ontology term is a node, and the relationships between the terms and the edges, the relationship between the terms are the edges between the different nodes. Gene ontology is loosely hierarchical, with child terms meaning more specialized than their parent terms. But unlikely in a strict hierarchy, a term may have more than one parent. Note that the parent-child model does not hold true for all types of relations. For example, the biological process term, hexose biosynthetic process, has two parents, hexose metabolic process and monosaccharide biosynthetic process. You're getting it like intuitively. So hexose biosynthetic, what is a hexose? Hexose is a monosaccharide, right? So its parent term will be monosacharide biosynthesis process. And also hexose biosynthetic process is the biosynthetic term. What is biosynthesis? It's metabolism, right? So hexose metabolism process will be a parent for this particular term. So similar kind of things are like basically similar kind of concept is followed in gene ontology. Is particularly is that particular gene involved in what particular process, and what is the parent process of that particular process. So it's a hierarchical it's a hierarchical network. But one thing to keep in note, that one particular child or a leaf can have two parents, two nodes, right? So yeah, this, if you want to go through, please do. I'll keep this in the chat. Okay. Sorry. So there are actually two different entities that are commonly referred to as gene ontology: the ontology itself and the association between the gene products and the gene ontology terms. But the term ontology or the gene ontology is commonly used to refer to both, which is sometimes a source of potential confusion. In order to avoid this, here we will refer to the term geo-ontology to describe the set of terms and their hierarchical structure, and geo annotations to describe the set of associations between the genes and the geoterms. There are three types of terms or domains in gene ontology, as I was saying: biological processes, molecular function, and cellular components. So this is basically a hierarchical structure and ancestry relationship in gene ontology. These terms that are in lower in location are basically lower levels, are more specific terms, while if you move higher up, those will become more general terms. Say for example, we start with regulation of programmed cell death, right? The parent term of that will be programmed cell death, the parent term for that will be cell death, the parent term for that will be a cellular process or a regulation of a biological process, and the parent term for that will be a biological process. So it is describing a hierarchy, right? So gene ontology analysis is something that is used for getting an idea about what is the different types of factors, be it biological process, be it a molecular function, or be it the cellular component. What is a set of genes actually contributing to in a cellular system? be it in a location wise, be it in a function wise, or be it in some other process wise. So why is this important here? Say for example, you get a set of genes which are differentially expressed in a particular condition. Now you want to see which pathways or which biological processes or which molecular functions are associated with these particular genes. So you do a gene ontology analysis. So you give a set of genes to the ontology browser, and it will give you a graph like this. So you'll have on the lowest end you'll have the most precise function for that particular set of genes, and if you go higher and higher up, you'll get a more generalized term, right? So yeah, so this is true for all types of ontology, not only gene ontology. I showed it with you with tissue ontology as well. So there are many types of ontology. It's just a naming system and a hierarchical process to capture the relationship between these separate terms. Okay. Now, okay, yeah. So now we come to these different types of statistical tests and how you can get you have identified a set of genes, and now how you can say that this set of genes are actually differentially expressed, or how is it actually affecting a particular phenotype or whatever it is doing. So one concept that is used here, couple of concepts. So one is GACA that we'll look into the next slides, and the other thing is the hypergeometric distribution. So I'll just want to give you an idea about what is hypergeometric distribution, then we'll look into how it is applied in biological systems. So in probability theory and statistics, the hypergeometric distribution is a discrete probability distribution that describes the probability of k successes, random draws for which object withdrawn has a specified feature, in n draws without replacement. So without replacement is a very important term here. If you do replacement, then it doesn't remain a hypergeometric distribution, it becomes a binomial distribution, right? So hypergeometric distribution is only when there is no replacement. If you say, for example, you are taking out, like you are given a set of balls in a bowl and you are taking out the balls one by one. After taking out, if you put the ball down before picking up the second one, that becomes a binomial distribution. But if you don't do that, you just take out one ball and keep it aside, and then you go, and now your sample size is n minus 1, that becomes a hypergeometric distribution. So the hypergeometric distribution tells you the probability of k successes in n draws without replacement from a finite population size and containing a set, like finite exactly this number of objects with that particular feature, wherein each draw is a certain success or a failure. So there can be nothing in between. So you are given, say, 5 colored balls, black, white, blue, red, green, and you are asked to pick out one. So if you are considering success to just get the red ball, then getting a blue or a green ball, those are failures. The system is managed that way, right? So and in contrast, the binomial distribution describes the probability of k successes in n draws with replacement. So in hypergeometric it's without replacement, and in the binomial distribution it's done with replacement. So this is basically 4 types of distributions which are like very close to each other. So binomial is sample n events with replacement. Doesn't matter when your success or if it's a success or failure. Geometric is sample events until one success happens, right? Hypergeometric is the same thing as geometric, just without replacing the sample size. Binomial is the same as a negative binomial, just adding the factor that there needs to be this amount of successes, which is not associated in the binomial distribution. This is the probability mass function of hypergeometric distributions. Yeah, this is the probability mass function of hypergeometric distribution. So your capital N is basically the total population size, your K is the total number of, say in this case, the red balls, and N is the number of tries that you are doing. So depending on the value of K, so K is basically the number of successes here. So small k, it's not this big K. the small k is the number of successes here. So based on that, you can have like distributions like this. So this is how a negative, a hypergeometric distribution looks like, right? Now, how do we implement hypergeometric distribution or a hypergeometric test for enrichment? So look at the right hand side first. So for example, given an urn of n marbles, capital K of which are red and capital N minus K are green. If we draw small n marbles, what is the probability of drawing exactly K red marbles? So this is basically, this Venn diagram is showing you. So you have, this is the total green plus red, right? This is your total sample size. Out of that, you'll have small k drawn red, small k drawn red balls, and you will also have red marbles, and you will also have this is denoting the total red marbles, this is denoting the drawn red marbles, and this is denoting the total red and green that are not that are basically present in the system. So according to this is basically it is equivalent to a one-sided Fisher test, and the formula is given by this. So it's exactly kind of like the binomial distribution formula KCI I equal to K to n minus K C n minus 1 divided by capital N C small n. So this is your hypergeometric distribution formula for capital N marbles. You have capital K red marbles, and you want to draw exactly small k red marbles, and you want to draw n marbles, and you want to find out the probability of, out of those n marbles, what is the probability of drawing small k red marbles, right? So same concept given in the right hand side is implemented in enrichment analysis as well. So given a signature, which is a list of genes or a set of pathways or whatever it is, test whether the signature has an over-representation of genes belonging to a given category, example a pathway or an ontology term. And the example is basically what would you want? You want, is my signature significantly enriched for DNA damaged genes, or is my signature significantly enriched for metabolic genes or whatever. So, and also if is my signature is significantly overlapping with any other kind of signature. So similar concept you apply to enrichment analysis as well. So in this case, this is your total population, this is the overlap between your k successes and your total number of genes that are present, and this is the formula that you use to give you the probability that that particular set of genes are actually overrepresented in your data set or not. So sorry, I think the light is low. Yeah, okay. So that was hypergeometric test for enrichment analysis. So you have a set of genes, you are checking if that set of genes is overrepresented or underrepresented, or they are belonging to a particular category or a set of pathways or whatever it is doing. So you kind of take the concept from hypergeometric test. Now we look at the similar enrichment analysis procedure known as GACL. This is one of the most commonly used techniques for enrichment analysis. And why is GACL needed? So detecting modest changes in gene expression datasets is hard due to various factors: the large number of variables, the high variability of samples, and the limited number of samples. So the goal of GACL is to detect modest but coordinated changes in pre-specified sets of related genes. Such a set might include all the genes in a specific pathway, on a specific chromosome, or on a specific cytoband, for instance. So it can be anything actually. You just want to check if I have a pre-specified set of genes. Is my data supporting that this pre-specified set of genes are somehow enriched in my data, right? Okay, so we'll go through it step by step. So the first step is basically calculation of the enrichment score. So first you rank the genes based on the correlation between their expression and the class distinction, right? So you do a kind of a Pearson or a Spearman rank correlation method, mostly Spearman rank, because it gives you a ranked set of features. So rank genes based on the correlation between their expression and the class distinction for an a priori set of genes. So basically you have a set of genes that you have pre-computed, then you take those genes and you compute the cumulative sum over all ranked genes. Yeah, you rank the genes based on the correlation between the expression and the class distinction. Then you have ranked, then you have a set of genes that you already know are like your basically pre-specified set of genes. You check those genes in that particular rank and you compute the cumulative sum of the expressions over the ranked genes. Increase the sum when the gene in S, when the gene is in S, and decrease it otherwise. So you are just doing a random walk. So you start from the top, you go to one gene by another. If a particular gene is present in that particular data set, in the ranking part, and it is also present in your own gene list, then increase, otherwise you decrease the magnitude, right? The magnitude of increment depends on the correlation of the gene with the phenotype. Excuse me. And you need to record the maximum deviation from 0 as the enrichment score. So you have a set of genes like this, you have a ranked gene list. Now, like all the genes in this list are not present here, so you have these lines are denoting that the genes are present here in the gene set. So what you do is basically you do a random walk, you add if the gene is present in the list, you subtract if the gene is not present in the list, and you get a distribution, or you get a value like, if you plot the values of all the cumulative sums, you get a distribution like this, which the top most maximum deviation from 0 is what gives you the enrichment score. So like you add, you subtract, you add, you subtract, you get the set of values, then you plot the values and you see which is the maximum value that you have reached. So that is your enrichment score. So that is step 1 of GACA. Now these are 3 examples. So this is basically X chromosome inactivation, this is another pathway, this is another NKT pathway. So you'll have a set of you already have a set of genes, you are checking the set of genes in your data set. You find that gene, you move up, next you find that gene, again move up, then slightly move up, then here you go and the gene is not there, you move down, then again the gene is there, you move up, like this you go, and you reach the maximum point here, after that which goes down completely. So this is your enrichment score here. But in this case, you can see there, like the vertical lines, which are basically these are the lines. These lines indicate the locations of maximum enrichment score and 0 correlation between expression and phenotype, right? So these are your maximum enrichment scores here, here and here as well. So you take the maximum out of all the maximum, right? So this is how we get the enrichment score. Next is the step 2 is to assess the significance, which is the nominal p-value, where you have to permute the phenotype labels 100 times. So you just do a randomized permutations of the phenotype labels, and then you compute the same enrichment score as above for each permutation. So you permute, like basically you just randomize the set, then you again do the same enrichment score analysis, and then you plot the scores and you see what is the total like highest enrichment. So you do that for like 1000 times permutation, and you compare the ES score as above for each permutation. Now you compare the EH score for actual data to distribution of the ES scores from permuted data. Permuting the phenotype labels instead of genes maintains the complex correlation structure of the gene expression data. And in the next step, you adjust for multiple hypothesis testing, right? So in the first step, you calculate the enrichment score. In the second step, you permute the genes and you calculate multiple enrichment scores and you assess the significance. And in step 3, you do multiple hypothesis testing. You normalize the enrichment score, accounting for size of each gene set, yielding normalized enrichment score, which is NES. So basically you divide the enrichment values by the total number of genes present in the gene set, that's all. And then you control the proportion of false positive by calculating false discovery rate corresponding to each normalized enrichment score by comparing tails of the observed distribution, tails of the observed and null distributions for the normalized enrichment scores. And this is the final step in the GSE algorithm. It's basically, in the original method, equal weights is used for each gene. In the revised method, what you can do is you can give weighted genes according to their correlation and phenotype. So basically, the correlation that you're calculating and ranking them, you're just not only ranking them, but you're using the correlation as a weight for calculation of the enrichment score. This may cause an asymmetric distribution if the enrichment scores, if there is a big difference in the number of genes highly correlated to each phenotype. Consequently, the above algorithm is performed twice, one for the positively scoring gene sets and one for the negatively scoring gene sets. Right? Okay. So yeah, that's all that I needed to cover. Yeah, that's all that I needed to cover for this week. And now we'll move into the assignment parts. So I'll keep the questions as I do. I keep the questions on screen for a couple of minutes or more, depending on the question, and then we'll discuss the answers, right? Okay. Just a second. Yeah. Okay. You can write the answers in the chat box, or if only one of you is answering, you might unmute yourself and answer as well. Okay, just give me a minute. I'll just be back. My water has finished. I'll just be back. Yeah, so okay, I'll give another couple minutes for other people to work out, then we'll discuss this. Okay, I think we have any answers. Okay, so let's discuss this. So the question is, you are doing multiple hypothesis testing. You have done 50 statistical tests, and the probability of making one type 1 error is given. And you are asked what is the probability of type 1 error for each statistical test, right? So how we calculate is basically probability of not making type 1 error. So if this symbol, I'm not sure why they have provided a symbol, but anyway, so this symbol represents probability of type 1 error for each. So probability of not making type 1 error will be 1 minus that, and to the power will be the number of statistical tests you are doing, in this case n, which is equal to 50 here. So the probability of not making a type 1 error is given by this 1 minus the symbol to the power n, and and the probability of making a type 1 error will be 1 minus this whole expression, right? And now you put it here. So you are given this value, the probability of making a type 1 error for multiple hypothesis is given as like 0.395. You put it here, your n equal to 50. Now you need to solve it, like 1 minus p to the power 50 equal to this value, and you take a log and then you solve it, and ultimately the probability will be coming as 0.011, which is correct. So the answer is option B. This is actually pretty simple. So just think about it like this. You are given the probability of making an error, but basically for one particular case, right? For one particular testing. So you are asked what is the probability of that particular type 1 error for one particular test. You are given that for the error for all the 50 tests. So you assume a probability for the type 1 error for each test, then you calculate the probability of not making an error, which is basically 1 minus, and for n statistical test it will be to the power n. Then you make 1 minus to get the probability of making of error again for the whole multiple testing. That value is given to you, then you solve it, and 0.011 is the answer. So yeah, these kind of questions will come in your exam, so I'll suggest practicing these a little bit in your free time as well. Okay, let's move on. Yeah, the answer is correct. So FWER I did not discuss much today, but sir has discussed in the course itself. So it's basically the concept of family-wise error rate as a probability of making a type 1 error among a specified group of family or family of tests, right? So Bonferroni correction is one of that we do. We just divide it by the number of tests. We divide the p-value by the number of tests to get the significance threshold. And Bonferroni correction is another one that we have not discussed, but yeah, you should know these, at least the name of the tests, right? Okay, moving on. Actually, FWER is actually used to reduce the number of false positives. So it's actually counterintuitive to what you're saying. So it's not true. But FDR allows a certain number of false positives. So like we take the FDR cutoff of 0.05, allowing at least 5% false positives, and yeah. And Born Ferroni correction shows more type 2 error than home born Ferroni correction. But FWER is the reason we have, basically it is the logic behind doing FWER is to reduce and completely get rid of false positives, right? So it's not true in that sense. Yeah. I know it's the options are a little bit closer to what we would have liked, but yeah. Okay. Okay. 4 by 95, I don't know. 44, 0.46. Okay. Let's see. Okay, so you are given ki in a bin there are 20 balls, and like out of this 20, 12 are red, 8 are green. If and you are asked if you are drawing 3 balls and random from the bin, what is the probability of being 2 balls from red and 1 ball from green? So here sampling is done without replacement, which essentially means you have to apply the hypergeometric test, right? So among 20 balls, 12 balls are from apoptosis pathway, which is red, and 8 balls are from oxidative phosphorylation, which is green. So probability of 2 balls from apoptosis pathway and 1 ball from oxidative phosphorylation pathway, if we draw 3 balls of random containing ball bin of 20 balls, is basically 12C2 is the number of times that uh, you can take 2 balls and get both of them red. So 12 to C2 representing that, 8C1 representing taking 1 ball from for green or oxidative phosphorylation. So you multiply that and you divide it by the total number of possibilities, which is 20C3. So if you do the calculation here, it comes out to be 0.586. So it's pretty simple. Like you take 2 balls from red. So how do you take it? 12C2. You take 1 ball from green. How do you take it? 8C1. Now how do you take all 3 together? You multiply, and then you divide by the total number of possibilities, which is 20C3. So the answer is 0.05. Okay. This is a fairly simple question. Do you have any answers? No, multiple hypotheses. Acha, in this way. Okay, acha, acha. Yeah. Yeah, the answer is correct. So for when you're correcting for multiple hypothesis testing, you are reducing the number of false positive and increasing the number of true positives. So yeah, your answer is correct. If you do multiple hypothesis testing, the number of false positives increase. Yeah, correct. Sorry, yeah, that's repetition. answers. Okay, the answer is gene set enrichment analysis. So here, as I was showing, if I go back to the slide here, um, yeah. So for GACA, functional class coding methods, um, can you please mute yourself? Okay, so for class coding methods like GACA, you need rank-based gene sets, right? It's a rank-based approach, so you need to have a list of genes with apparent ranking order. So unless and until you have that, it's not appropriate to use GACA or any FCS approach, right? So let me go back here. Yeah, the correct answer is gene set enrichment analysis. Okay. Okay, gene ontology, it's a database of functional annotations, biological processes, molecular function, and cellular like locations of the processes is also incorporated in that, and it's part of one of the sub ontologies, and it's also partially hierarchical. So, and the last option is actually opposite. The daughter nodes describe more specialized functions than parent nodes. So the D option is not correct. So ABC is the option. Moving on. here. Um, uh, Hi, Dr. Srividya. But yeah, no, molecular functions is included in one of the subcategories, and these set of questions are not actually from the assignment. So these are basically older assignment questions that we discussed. This is not from your current assignment. These are not supposed to match with your current assignment. So yeah, and yeah, molecular function is a subcategory as I showed in this particular slide. Let me go back. Yeah, biological processes, molecular function, and cellular components. These are the three main like subcategories, sub-ontologies in gene ontology, right? So, and the lower nodes or the daughter nodes give you more information than the parent nodes. So, okay. So the answer to this is MSigDB and KEGG. KEGG is a pathway related database, and MSigDB is also the same. So I'll just quickly show you. Uh, KEGG is the pathway database. Here you can get, so for example, you take this particular pathway, let it open. Yeah, so it gives you the pathway information and the genes that are involved and everything, which pathway affecting which. And MSigDB is the database for GSA, right? So yeah, from here you can see that these are the hallmark gene sets. So you have like all the these set of gene sets are right this, all gene sets you can browse from here, all the pathways are given here, and these 50 are your these are your basically the gene sets that are present here, and these are only like a subset of those sets, and I'll just quickly show you how the gene sets look like. So if you download this in a GMT format, so for GSA we generally use the GMT format. So yeah, so it gives you, this is your pathway name, this is basically the source, and after that these are your all your genes. So you can get your gene sets and everything from MSigDB and do your analysis. So yep, these two databases are used for like functional enrichment. Okay. Yeah, the correct answers are so a score decreases if a member of the gene set pathway is not found while traversing through the ranked gene list. So what you do is you pass through each of the genes, and if the gene is present in your gene list, you increase the score. If it is not, you decrease the score. So that is correct. And the final ES score is the maximum deviation from zero, as I was saying in the slides as well. So you do this for all the genes, then you get the values for enrichment scores for like after you have passed every gene, and then you plot that and you check which is the maximum enrichment score that has been reached for this particular set of genes, and that is your maximum so that is your enrichment score, right? Yeah. Okay. Okay, let's see the answers. Let's see it. Okay, it's a pretty simple question actually. The answer is both are true, both the assertion and the reason are true. So why we normalize is basically when you're comparing enrichment scores from two different gene sets, the gene set size is an important thing to consider, because more the number of genes in your gene set, the higher probability that you will reach a higher enrichment score for that. So that is why you normalize the enrichment score by the total number of genes that is present to get the normalized enrichment score. So that's how you can, like, if you take the normalized enrichment score, if you have two different gene sets with two different set of genes with like the numbers also being different, like in one set you get 15, another set you have 30, but after you normalize with the gene set, you can directly compare the values of the enrichment scores. So that is why the normalization is important. So yeah, both are true. Okay, that's it from my side. And yeah, if you have any questions, you you might ask. And yeah. Okay then, if there are no questions, I'll close this session. Thank you for joining, and see you next week, same time. Thank you.